Today we will be learning about DNA replication and the enzymes involved in DNA replication. These are the enzymes involved in DNA replication: DNA polymerase, helicase, primase, ligase, DNA topoisomerase, and SSB protein. Now let us look at each of these enzymes one by one. So, DNA replication always starts with the help of helicase enzyme. Then, DNA polymerase works on the synthesis of the strand. This DNA polymerase only works with the help of a primase. Then, the Okazaki fragments are being glued with the help of ligase enzyme. Then, the topoisomerase enzymes are involved in reducing the strain in the double helix of the DNA strand. And then the SSB proteins, which keeps the two strands away from reforming again into a double strand. Now, let us look at the process of DNA replication. Then only we will be dealing with the definitions of these enzymes. First one, as I told you, that DNA replication always starts with the help of an enzyme called helicase. So helicase enzyme always help in the unwinding of the double-stranded DNA structure. Now, before looking at the unwinding process, let us see the polarity of the DNA strand. We know that the DNA is a double-stranded structure which has two ends, the 3 prime end and the 5 prime end. Also on the opposite strand there are 5 prime and 3 prime. So this is what we call it as the polarity of the DNA strand. Two strands anti-parallel direction. So this is a double helix DNA structure. When we uncoil it, we'll get a structure like this and which have opposite polarities, polarity difference. Now let us look at the functions of each enzyme one by one. The first one is helicase enzyme. Helicase is involved in the unzipping of the DNA helix, as I told you. So they break the hydrogen bond between the base pairs. This is a double-stranded DNA enzyme which has hydrogen bonding in between the base pairs. These are the base pairs in between which there are hydrogen bonding, which is being broken by the help of helicase enzyme. So it is actually a unwinding enzyme which works on the DNA strand. Now let us look at the DNA polymerase enzyme. DNA polymerase are classified into three: DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Each of their function is actually the synthesis of base pairs. DNA polymerase 1 helps in the removal of DNA primers and replaces them with the DNA. DNA polymerase repairs the DNA damage, and DNA polymerase 3 synthesizes new DNA strands during replication. Now I'll be explaining the whole process and the uses of each enzyme on the replication. See, this is the function of a DNA polymerase. So DNA polymerase and primers act complementary to each other. That is, for the DNA polymerase, the main condition is that the DNA polymerase So DNA polymerase has a condition. Number one condition is it will bind only from 5 prime end to 3 prime end. Also, it needs primers for its initiation. So these are the two conditions. Second condition is requires primers. for the process of initiation. Now, when we look at the whole process, since I told you DNA polymerase is for the synthesis of the new strand. So another strand is being synthesized over here like this. So the primers come and fix a these are the primers which come and fix a position somewhere here, and then this is the DNA polymerase. This is the DNA polymerase, and after the primer sets a position on the strand, the DNA polymerase start the synthesis of the base pairs. So as I told you, DNA polymerase can only synthesize from 5' to 3' strand. So in this case, we have got a double-stranded DNA with the polarity difference. That is, here it's 3' and on the opposite it's 5'. Here it's 5' and on the opposite it's 3'. But when it comes to the another strand, it is a problem, because when the primers is being set over here, the primers is set somewhere over here, what happens is, what happens is that since I told you that the DNA strand could only bind from 5' to 3'. So when a primer is being set over somewhere over here, and then the DNA polymerase starts its activity, it will create a polarity of 5' to 3'. But here when you see, you won't find a polarity difference in this strand, since see, this is 5' and this is also 5'. So we cannot start the initiation from here, from this end. So the primers start from somewhere else. So primer is being set over here for the DNA polymerase to start its activity. So it will start from here and 5' to 3'. So here when we see, we find a polarity difference. Okay, this can be done. Now when the primer turns back, they find that the helix is opening the double helix structure of the DNA. So again another primer is being set over here and DNA polymerase starts synthesizing from 5' to 3'. So in this way we find the fragments are being created over here. See, this is one fragment, this is another fragment. In such a way many fragments are being created. So this forms a discontinuous strand. Now we call this discontinuous strand as the Okazaki fragments. These kind of discontinuous strands are being generated. And here, as I told you, primers will be there. Okay, these are the primers. Now what happens is, this discontinuous strand is being made continuous with the help of another enzyme called the ligase enzyme. See, this is a discontinuous strand and has to be joined and made into a continuous strand with the help of another enzyme called ligase. In case of this strand, it has been perfectly generated since the polarity was perfect. Like it was starting from 5' to 3' end, and it generated a polarity difference in the strand, newly formed strand. But when it comes to this strand, we cannot start from this side. So primer started from somewhere in the middle and then started the DNA polymerase synthesis from this end to this end. And then when the primers turned back, they found that the helix has uncoiled the double-stranded DNA. So again they set a position somewhere here and started binding. Again the helix was open and primers came here and synthesized like this. So it formed a discontinuous type of strand. So this is being joined with the help of another enzyme called ligase. So ligase is the enzyme which joins these strands. The primers are being removed and a continuous strand is being formulated. Now look at the function of the DNA polymerase 1, 2 and 3. Now you'll understand after reading the lines. See, DNA polymerase 1. removes RNA primers and replaces them with DNA. Fine. RNA primers were being set down in the strand and are removed by the DNA polymerase so that base pairs could be synthesized over there in place of the primers, right? Now, DNA polymerase 2. They repairs the DNA damage. If any damage or any base pair is missing from the place, then DNA polymerase repairs that one. Now DNA polymerase 3 synthesizes new DNA strand during replication. So as I told you, in the formulation of this strand there is no problem. So DNA polymerase 3 is working over here. Okay, so these are the main functions of DNA polymerase 1, 2 and 3. Now the primers, as I told you, they are the initiation point provider or the starting point provider for the DNA polymerase to start the synthesis activity. Now the enzyme ligase, as I told you, the fragments generated were called the Okazaki fragments. They are the Okazaki fragments. Okay. So Okazaki fragments are being joined with the help of ligase enzyme. So you can see over here on this side, when you see, you find a continuous strand. This is a continuous strand, but here the another strand is discontinuous with Okazaki fragments present over here. It is a fragmented strand and is being ligased or glued with the help of enzyme called ligase. Ligase is an enzyme which glues the whole DNA strands. Now this is the definition of the ligase. This is the sealing DNA replication. So ligase is an enzyme that plays a crucial role in DNA replication by joining together the Okazaki fragments. Next one is topoisomerase. As I told you, topoisomerase reduces the strain on the double-stranded helix. This actually happens in this way. This is a double-stranded DNA helix which is being uncoiled with the help of helicase enzyme. So when it uncoils in this direction, what happens is a kind of supercoiling is being present. Supercoiling happens as the activity continues in this direction. So to reduce this supercoiling, this topoisomerase helps in reducing the strain when this helicase enzyme works in this direction, and then further on moving on, it creates a supercoiling effect which is being reduced with the help of topoisomerase. Now the SSB proteins, keeping DNA straight. We know that the double-stranded DNA is being opened with the help of an enzyme called helicase, and this double-stranded DNA remains separated with the help of enzyme called SSB protein. We know that this strand would particularly have a tendency to form as if they were, that is double-stranded. So the helix activity would be waste. So to avoid that, SSB proteins help in keeping the strand separated throughout. Yes, that's all. Thank you.